Hi, my name is Sarita, lecturer in Botany, Government Degree College for Women, Nalgonda. Today in this video, I am going to talk about Pinus needle anatomy and its xerophytic characters. This Pinus belongs to division Gymnosperms, class Coniferapsida, order Coniferales, family Pinaceae, genus Pinus. Pinus dwarf branches, that means limited growth branches bears needle-like chlorophyllous leaves. Pinus dwarf branches bear needle-like chlorophyllous leaves. This Pinus needle shows many xerophytic characters. Only midrib is present, lateral veins are absent. Only midrib is present, lateral veins are absent. Transfusion tissue replaces the loss of lateral veins. Transfusion tissue substitute the function of lateral veins. In transverse section, Pinus needle shows triangle in shape, semicircular in shape, circular in shape. Pinus longifolia shows semicircular in shape. Pinus sylvestris shows circular in shape. In transverse section, Pinus strobus shows triangle in shape. The three main parts seen in the needle, they are epidermis, mesophyll and vascular cylinder. Transverse section of the Pinus needle shows mainly three parts, they are epidermis, mesophyll and vascular cylinder. Coming to the epidermis, this is the epidermis. This epidermis is the outermost layer, 1 cell thick without any intercellular spaces. It is covered by thick cuticle layer and it contains shrunken haplochelic type of stomata. Shrunken haplochelic stomata are present in this Pinus needle. Haplochelic means guard cells. Subsidiary cells originate from different initials is known as a haplochelic type of stomata. Here we can see haplochelic type of stomata. This is about epidermis. Epidermis is the outermost layer, 1 cell thick without any intercellular spaces. It is covered by thick cuticle layer. It contains shrunken haplochelic type of stomata. Next, coming to the hypodermis. Below the epidermis, hypodermis is present. This is the hypodermis. Below the epidermis, hypodermis is present. Below or beneath the epidermis, 2 to 3 layered hypodermis is present. It is made up of sclerenchymatous tissue. Below the stomata, air chambers. Below the stomata, air chambers are present in the hypodermal position. This is about hypodermis. Below the epidermis, 2 to 3 layered hypodermis is present. It is made up of sclerenchymatous tissue. Below the stomata, air chambers are present in the hypodermal position. This is about hypodermis. Next, move to mesophyll. This is the mesophyll region. Below the hypodermis, mesophyll is present. It is made up of parenchymatous tissue and not differentiated into palisade and spongy tissue. Mesophyll cells have thin cell wall and this mesophyll shows finger-like infolding structures to increase the surface area for photosynthesis. Mesophyll. Below the hypodermis, mesophyll is present. Mesophyll region is present. It is made up of parenchymatous tissue. These cells not differentiated into palisade and spongy tissue. Mesophyll cells have thin cell wall and this mesophyll cells shows finger-like infolding structures to increase the surface area for photosynthesis. This parenchyma is known as a armored parenchyma. Next, this mesophyll contains number of resin canals. These are the resin canals. These resin canals lined with epithelial cells. This epithelial cells secrete turpentine into the resin canals. Next, last layer of the mesophyll is known as a endodermis. Limiting layer of the mesophyll is known as a endodermis. This endodermis contains barrel-shaped cells and prominent Casparian strips. This is about mesophyll. last layer of the mesophyll is known as a endodermis. It contains barrel-shaped cells and prominent Casparian tips. This is about mesophyll. Next, move to vascular cylinder. This is the transfusion tissue. This transfusion tissue present in between this endodermis and vascular bundles. This is the transfusion tissue which is present in between the endodermis and vascular cylinder. This transfusion tissue substitutes the function of lateral veins. This transfusion tissue substitutes the role of lateral veins. The transfusion tissue consists of 4 different kinds of cells. They are parenchyma cells, albumin cells, and transfusion tracted cell, and T-shaped sclerenchyma. This transfusion tissue consists of 4 kinds of cells. They are parenchyma cell and albumin cells, transfusion tracts, and T-shaped sclerenchyma. Coming to the parenchyma cells, which are present in between the 2 vascular bundles and endodermis. These parenchyma cells help in transport from vascular bundles to mesophyll. Next, albumin cells. Parenchyma, these are the vascular bundles. This is the phloem part, this one is xylem part. Parenchyma cells which are present near to the sieve tubes with starch and resin, that is turpentine. These cells are called albumin cells. Parenchyma cells which are present near to the sieve tubes with starch and resin. These cells are called albumin cells. These are the albumin cells. Next, transfusion tracts. Xylem cells which are present near to the vascular bundles. The xylem cells, lignified cells with thick walled dead cells with border pits and radial walls are called transfusion tracts. Transfusion tract cells, xylem cells which are present near to the vascular bundles. These xylem cells, lignified cells, and thick walled dead cells with border pits on radial walls are called transfusion tract cells. These are the transfusion tract cells. Next, T-shaped sclerenchymatous tissue. T-shaped sclerenchyma present in between the 2 vascular bundles. Next, move to vascular bundles. 2 vascular bundles are present at the center of the vascular cylinder. These vascular bundles are conjoint, collateral, open, and endarc type. Open means in between the phloem and xylem. KMM is present. That type of vascular bundle is known as a open type of vascular bundles. Here, conjoint, collateral, open, and endarc type of vascular bundles are present in this Pinus needle. This is the phloem part. This phloem contains sieve tubes and phloem parenchyma. Companion cells are absent. Albumin cells substitute the function of companion cells. This is about Pinus needle anatomy. Next, move to Pinus xerophytic characters. Needle-like leaves are the one of the xerophytic character. Presence of thick cuticle layer is the one of the xerophytic character. This is the cuticle layer, also one of the xerophytic character. Shrunken stomata in epidermis, it is also one of the xerophytic character. Sclerenchymatous hypodermis is also one of the xerophytic character. Presence of transfusion tissue in the leaf, it is also one of the xerophytic character. Transport of organic substances between the vascular bundles to mesophyll through transfusion tissue, it is also one of the xerophytic character. Albumin cells transport organic substances between the phloem tissue and mesophyll, it is also one of the xerophytic character. This is about Pinus needle anatomy and its xerophytic characters. Thank you.